At Amherst, David was part of the Independent Scholar Program. He went on to earn a law degree at the University of Chicago before earning his medical degree from Harvard Medical School. Welcome, David, and thank you for being here. Dr. Harold Varmus, class of '61, was the co-recipient in 1989 of the Nobel Prize in Physiology or Medicine for studies of the genetic basis of cancer. He is the Lewis Thomas University Professor of Medicine at the Meyer Cancer Center of Weill Cornell Medical Center in New York City. He's also Senior Associate Member of the New York Genome Center, where he helps develop programs in cancer genomics. He's also Adjunct Professor at the Macaulay's Honors College at the City University of New York. Harold was Director of the National Cancer Institute for 5 years. He was President of Memorial Sloan Kettering Cancer Center for 10 years and Director of the National Institutes of Health for 6 years. He serves on numerous advisory groups for academic, governmental, philanthropic, and commercial institutions. He's authored hundreds of important scientific papers and 5 books, including a 2009 memoir entitled The Art and Politics of Science. At Amherst, I'm pleased to say, Harold Varmus majored in English, and he went on to receive an MA in English Literature from Harvard University before deciding to go into medicine at Columbia. Hosting and contributing to today's conversation is Amherst College trustee Shirley M. Tilghman, who is an honorary degree recipient from Amherst College in 2008. She's Professor of Molecular Biology and Public Affairs and President Emerita of Princeton University. Shirley was a founding member of the National Advisory Council of the Human Genome Project for the National Institutes of Health. She's received many, many awards as well, including a Lifetime Achievement Award from the Society for Developmental Biology. She serves on the Science Advisory Board of the Chan Zuckerberg Initiative. She's director of the Broad Institute of MIT and is a fellow of the Corporation of Harvard University. On Tuesday this week, just 2 days ago, she was selected by New Jersey's Governor, Phil Murphy, to co-chair the state's Restart and Recovery Commission, overseeing the timing and process of reopening the state after pandemic-related closures. It's a great honor and a gift to have you all here. Take it away. Shirley.
Thank you, Biddy, and good evening, everyone who are listening to this and watching this podcast. I'm going to just launch right into some of the questions that I think are on many people's minds that our 2 experts have a great deal to say about. The first, I'm going to direct Harold. This is not the first time that we heard the word coronavirus. What did we know about coronaviruses before this pandemic, and what do we need to learn about it now?
Well, I think, Shirley, many people were surprised to learn that this so-called new virus was actually a member of a large virus class. There are hundreds of isolates that correspond to the term coronaviruses. They're named, as I think many people have recognized now because of the glorious pictures that are on the NewsHour and other places, that it's coronaviruses have what looks like a crown of thorns around their entire spherical body. Those spikes on the surface that you see next to Judy Woodruff every night are proteins that the virus uses to enter cells. These many viruses inhabit a wide number of host species. They're not only found in human beings, although we know of at least 4 such viruses that have been causative agents of common colds as known for many years. But these viruses, as I think probably all of the listeners are aware, can be found in bats and pangolins and camels and tigers and many other animals. So that's one of their extraordinary features, and it makes them, at one level, somewhat akin to the influenza viruses. We're also found in many species, and indeed their ability to hop from species to species and then infect human beings is one of the reasons they pose a risk to us. And indeed, there have been at least a few other outbreaks, most famously the SARS outbreak and the MERS minor epidemic several years ago, that have introduced coronaviruses to the general public. This one is different from the others in many regards. When you look at an evolutionary tree, you find that it's incredibly closely related to the SARS virus that was found in Southeast Asia in 2003. So a lot of what we've learned about this virus class has been one of many ways that science has prepared us for dealing with this epidemic. And I'm sure in due course we'll get into the issues of what that long history of studying coronaviruses has meant for this epidemic.
And yet, what I'd like David to comment on is despite what Harold has said, it seems that both the SARS and the MERS epidemics did not sort of produce the kind of canary in the mine alert sign that would have made us better prepared for this. Why do you think that is?
I think if you look at the epidemiology of both MERS and SARS, the fatality rate, the degree of infectivity, the human-to-human transmission, sort of waned after the initial outbreak. I mean, I went back yesterday in talking to the people who are developing monoclonal antibodies against COVID-19. The good news is there's a lot of experience, and they were working and doing these monoclonal antibodies. And this is true also, certainly for the Oxford group on their adenovirus vector vaccine. They were doing trials in the Mideast, and there wasn't enough epidemic to even continue to test those agents effectively. And it didn't really hit us. I think if you ask ask Harold, what people were really worried about, the virologists that I know and talk to, I mean, they were losing sleep. And they expected a pandemic to come, but it was going to be influenza. It wasn't going to be coronavirus. I think that mistake or that lack of insight, because when we saw the coronavirus, people were really focused on influenza being the overwhelming virus and not coronavirus.
Harold, you wanted to add something.
I'm going to go back to your question about preparedness. And there are really two kinds of answers here to deal with this question of the difference with SARS and MERS. The first has to do with this, with the question of anticipation. And David's right that there's been more concern about flu in general in most virological circles, but we all recognize that there are lots of viruses out there that can cause a lot of damage. And it's not just flu that keeps most virologists up. That question of highly virulent viruses like Ebola, the recurrence of coronaviruses, because we know there have been a lot of coronavirus epidemics. But one of the things that society has failed to do, despite repeated warnings from scientists every time we have a pandemic, is to get our surveillance case together. We have incredible tools now for looking very widely at what's in animals, what's in the environment, what is the causative agent of some unexpected illness occurring in some remote place. There are so-called high throughput ways using remarkable sequencing technologies to look anywhere that you see signs of disease or unusual virus spread and find out what's out there. And this surveillance mechanism, which wouldn't be incredibly expensive, has been proposed over and over again. We've not done anything about it. The other question, though, the other side of this question is, how is this different from MERS and SARS? And one very important thing that I think we'll come back to has to do with the course of infection. And I think it's important to separate infection from disease, because especially in the case of this virus, infection is followed by several days in which people don't feel symptomatic. Maybe 2 days, maybe 6 days, maybe 28 days. And during that period, people have a lot of virus, a very high virus titer, as we refer to it, in the upper respiratory tract, in the nose, in the mouth, in saliva. And people can just be speaking and they spew out droplets that some of which are very small and can float very slowly. Vectors of transmission in a way that I think is quite different from what was true of MERS and SARS, which tended to cause disease more quickly without this prelude period in which people are feeling pretty good and transmitting virus.
And therefore, because of what Harold said, the infectivity, is less, right? Because it's that asymptomatic period puts everyone at much greater risk.
Yeah, the infection is high. Yeah.
Yeah. So the question I was going to ask David is how unusual is this asymptomatic period for this coronavirus? It seems to me that there are two curiosities here for someone who's not an epidemiologist or a virologist. One is the degree to which asymptomatic people seem to be contributing to the spread of the virus. The other is the invulnerability of children. With influenza, the two populations that are most vulnerable are the very young and the very old. Whereas with this virus, it seems that the very young are relatively protected from infection. Do we know anything about that?
So let me take the children as a pediatrician, and Harold as a virologist can maybe talk about viral shedding and being asymptomatic. Shirley, I don't think we know yet the full story of how this affects children. There are two very conflicting views today. Switzerland, and please don't do this. For the grandparents who are listening, please don't follow what I'm about to say, because I have some concerns about it. But the chief epidemiologist in Switzerland says it's okay to hug your grandchild if that child is under 10. Now, they did not encourage family gatherings. The German epidemiologists jumped on that. And I don't think that doesn't quite make sense to me. We are starting to see, and we haven't worked this out, in young children, this Kawasaki-like disease that we still, that's basically a hyperimmune stimulated disease in young children that we think may be associated with this virus. And that we're just beginning to see cases, certainly in Italy and elsewhere. And we haven't worked that out. So I think the answer is we don't fully understand the story when it comes to children.
What's Kawasaki disease?
Harold, you want to?
No, you go ahead, David. You raised the issue.
It's basically, it has greater concerns. Because if you, it is I think what you see, it's a constellation of symptoms that you do see after certain viral illnesses that are not the virus, but the actual immune stimulatory effects that are associated with this virus. What this, and again, I think it's very early, but I think when you look at coronavirus, I almost see this course of this disease as two phases. And again, I think it's very early, but it's very important for how we're going to therapeutically approach this disease, right? You talked about the asymptomatic period prior to the onset of symptoms. But even with the onset of symptoms, you have certainly this viral phase of fever and cough. And then sometime around day 8, 9, 10, sometimes the second week, in some individuals, they crash, right? They have increasing O2 requirements. They have difficulty increasing shortness of breath. They end up being intubated put on a ventilator, many of them several days later. And we don't fully understand this second phase of this disease, because it's the second phase of this disease that overwhelms the lungs. Is it the reaction of the lung tissue to the virus? I mean, is it a viral toxicity? Is it a hyperstimulation of the immune system? Is it due to clotting? Is it due to even the ventilator? We haven't sorted this out. But the good news, and the real good news, because it was certainly reminiscent to me going back 30 years, when we've had the first drugs on HIV, the announcement yesterday on remdesivir that showed biological activity, clearly that's the first antiviral. I mean, and that will work, and hopefully we'll find other drugs for other targets and combination therapy of maybe multiple antivirals, maybe likely that they work early. But we also are going to need to have drugs for this second phase. And again, I don't think we know the mechanism. It may be IL-6. I mean, and we will. So I think we have to think about combination therapy and understand the course of this disease.
Yeah.
So
Harold.
Before we get back to therapy, which I think we should discuss in somewhat more detail, let me get back to your original question for this segment, Shirley, about kids. There are 2 points I'd like to make. The first is that we simply haven't done enough testing of people who don't have symptoms because of the shortage of available tests for the virus to know the answer to your question about why kids clearly have less of the now garden variety version of COVID-19. We just haven't tested that many children to see whether they're getting infected less frequently. They clearly have less clinical disease, but why that is not apparent. The second issue has to do with things that David was speaking about, and that has to do with our failure to understand fully the pathogenesis of this disease. We still don't know, because we haven't tested enough people, how many people actually get sick. We don't know why certain people get sicker than others. We know that there is a clear age dependency, that the older you are, the more obese, the more likely that you are to get severely ill. There are a number of other so-called comorbidities, hypertension, diabetes, and a few others. But what's even more interesting and striking is that there are a number of aspects of this disease which are totally unexpected. You can see in people who seem to be virtually asymptomatic, diffuse shading of the lung fields on chest x-ray. We don't fully understand what's going on there. People are frequently experiencing low oxygen, as David mentioned, without feeling short of breath, because they're getting rid of their carbon dioxide because of the way this virus seems to affect the lungs. And then there are a lot of other manifestations of the disease, including strokes and circulatory insufficiency in several organs, disorders of the kidney that are apparently caused by this virus or by the immune response to it that are very unusual. So we just don't understand enough about why kids in particular don't seem to have disease. And frankly, we don't know what the epidemiology of infection is. And it's important in this conversation to make a clear distinction between being infected with the virus and producing virus and being a carrier, and actually having symptoms and being said to have COVID-19, the disease.
Yeah. I mean, an issue that clearly is dominating the national discussion about this pandemic is the need for intensive testing. So the question I would ask both of you is, why in the most scientifically advanced country in the world are we finding it so challenging to develop the kind of testing capacity that we need to do the two things that I think both of you have been talking about, to identify who has the disease, who is carrying the virus, and who is carrying it in the population with asymptomatic.
Let me start on this one. And I think the word testing, first of all, has to be modified in every case by what you're testing for. So when we say the word testing, what most of us are referring to is testing for the virus. And But there is a whole other category of testing that David and I will both want to say something about, which is testing for a host response to the virus, looking for antibodies that say, yes, this person has been infected at some time in the past. So when you're testing for virus, traditionally in virology, and I think this is a teachable moment, as they say, for letting people who are dialed in today to know something about the history of testing for virus. But what was done traditionally is you go to the site of disease, the lungs, the respiratory tract, and you see if there's a living particle we call a virus, a particle which can infect cells and destroy them, copy, and multiply itself. No longer do we do that routinely. What we do now is we look with much more sensitive techniques that have been developed over many years of being molecular biologists, as all of us are. And that involves simply being able to detect the so-called chromosome of the virus, which in this case is a very long strand of RNA. And we have methods, some of them are familiar terms like polymerase chain reaction, PCR, and those methods allow us to see even very, very small numbers of virus particles that contain the viral genome. The reason we don't have enough testing going on is testing for virus is incredibly important. It tells you who's infectious, who might be able to transmit this virus to somebody else. And we don't have enough, not because we didn't identify the virus quickly. Indeed, you compare the speed with which we found this virus just a couple of weeks after the first cases appeared, and the speed with which we had a complete count of the nucleotide sequence of the genome, just a couple of weeks, extraordinary. But there was a defect in the supply chain, the development of tests. People were being unreceptive to the idea of using someone else's test. And we really failed to understand the epidemiology of this disease, despite this incredibly powerful set of tools we have for being able to test for the viral genome and say that there are virus particles there. We'll come back to the serological testing, the testing for antibody later.
So I think that if you look at the root of the delay, why did we lose a month or 5 or 6 weeks until we were able to get testing to begin to get ramped up, is that I think that if you look what happened, right? CDC decided, as it does many times, not to take the WHO test, and they were going to develop their own diagnostic. I mean, FDA went in and saw from the beginning that there were contamination problems with the way CDC was making that diagnostic. So when it sent out these test kits, they didn't work. There were problems. And I think both agencies really didn't get the urgency, certainly in February and even early March. And it didn't, I mean, these are generally, certainly, my old agencies are pretty conservative, small c scientific agency. It likes to get things right. It said to CDC, there were problems, but no one got the fact there was, the house was on fire. And it took weeks for someone to say, hold it, we have a national emergency. Now, once that happened, things shifted. But I really think that we underestimated, certainly CDC and FDA. Now, there were those, if you look at the emails that have come out, there were those who were literally screaming at the top of their lungs in their emails that this is coming, shut down. But I think CDC and FDA for that 5-6 week period before they figured this out were just flat-footed, and I regret to say that.
So how confident
understand why testing is so important? Can I just just go over, make sure everyone understands why we're and let's stay with what Harold's talking about, testing for the virus itself. In the absence of a vaccine, in the absence of a therapy, right? What is this about? There's an infected person, there's a susceptible person, right? And there's an effective, for the virus to be transmitted, there needs to be an effective contact, right? The only tools we have, I mean, really when it comes down to it, is can we reduce the number of infected people? Well, if they're asymptomatic and they don't even know they're infected, you got to test them in order to, you take them out of the system. But you got to, in order to take them out of the system, to quarantine them or isolate them, right? You need that test. The other tool we have is just to reduce the contacts we have with each other. In the absence of a vaccine, that's all we have. Now, those two tools, you know, for the last 200 years are very effective public health tools, but you can't reduce the number of infected people without testing. Now, as much as it was a mistake not to have testing early on, right? I mean, and we became overwhelmed because we really weren't able to contain this virus early on. We didn't have the testing to take those isolated cases and shut them down, right? Now, testing has become equally important, right? Because we have no idea how many infected people there are out there. And I don't know how you open up. I mean,
you've lost you, David. You're, someone's sending you a signal you've talked too long. I didn't do it, not me.
So while we try and get David back, David has said the magic word vaccine, which of course is something that is on everybody's mind. But before we talk about the vaccine, I think it would be very important to talk about what do we know about the human immune response to this virus that would suggest that a vaccine would actually be successful. Harold.
No, well, this is an incredibly important, maybe the most important question we can discuss tonight. So let me say, first of all, that we are deeply ignorant of the potential for making a vaccine here. But because we need to recognize that although we know there are antibodies produced in response to this virus and to infection by it, we don't actually know whether those viruses have the potential in all cases to either reduce the effects of a virus infection or to prevent infection entirely, which is the common principal objective of making a vaccine. But there are some encouraging signs. First of all, we know that some patients who have actually have antibodies that neutralize, that inactivate the virus. And we know this because we can take the serum from a person who's been infected, and we can show that that serum will prevent the virus from infecting cells in laboratory culture. So that's encouraging. Secondly, people have identified specific components of those antibodies and shown that they are highly effective in blocking infection. Third, people have done experiments in primates, other non-human primates, showing that antibodies can protect against infection, and then you can actually make a vaccine that works in rhesus monkeys that protects against an infectious challenge. We also can observe that patients do clear the infection. Now, the immune system is doing a lot of things, as David has already mentioned. We know that the immune system appears to play a pretty intense role late in the disease when the disease is particularly severe. We don't know exactly what the lethal components of that reaction might be, but it seems to be a reaction that includes some attack on the virus itself, because virus titers become very low, and eventually people who've had the infection and have recovered are free of any evidence of carrying live virus. So all those things point to the idea that the immune system can control this infection. Now, there are two questions. How well does it do that? How long does the recovered patient remain resistant to reinfection? And those are really important things we need to learn, because they're going to inform, how we approach vaccine development. And secondly, they're going to give us an idea about how long we're going to be dealing with the virus called SARS-CoV-2. Some epidemics go away and we don't hear about the virus very much again, if at all. In this situation, we just don't know whether we're going to be seeing this virus, SARS-CoV-2, or some closely related version of this virus that's undergone a few mutations, call it SARS-CoV-3 and 4 and 5, in subsequent years, because the immune system doesn't have a good memory for this kind of agent.
So Harold, are there, urgent experiments that need to be done to develop answers to those questions as the vaccinologists are developing an effective vaccine?
There are huge numbers of questions, some of which could be approached simply with the tools that we already have. Questions we've been thinking about, such as the true transmissibility, the true prevalence of the virus. And what we need to do there is scale up the availability of tests. Now, we have at this point pretty good system for testing people who have symptoms to see if they have COVID-19. But we do have methods, thanks to a number of amazing developments in the world of genomics and other kinds of molecular biology, some remarkable new ways to go about screening large numbers of patients who seem perfectly well, to find out how many people in the population in New York and in Sioux Falls and in California have actually been exposed to this specific virus. And I hope that we'll be able to sort that out pretty soon. Secondly, we're going to be able to do the testing on such a large scale because of things that have happened in worlds very different from studying infectious disease, the worlds of genomics and so-called high throughput DNA sequencing, where machines have made it possible to test literally millions of people a day, and to get a much better picture of how much of the population is infected and able to transmit virus. With time, we'll be able to do large-scale tests for antibodies that tell us whether people have actually been infected in the past. And we'll eventually get a picture of the demographic of this virus that will allow us to feel more confident about whether we should be opening up our economy.
So one of the most recent studies was done by the New York Public Health Department that did a survey of 3,000 New Yorkers. Yeah, and discovered that 20%, roughly 20% of the population, of a random selection of the population within New York City, showed signs of having been exposed to the virus. Upstate, it was much lower. That's hardly surprising, given that the city is the epicenter of this pandemic. How, as a New Yorker, Harold, how does 20% feel to you?
Well, first of all, I have to question the result a little bit. It's not entirely clear that the test was perfectly accurate. There have been a lot of tests put on the market because the FDA agreed that these tests could be used fairly widely without being subjected to the kind of evaluation that they normally would be subjected to. And many have found that many of these tests are unreliable, with large numbers of false positives and false negatives. Secondly, the population of 3,000 that were selected for testing were not a totally random population. They were people who were asked to submit to the test. We hear you now, David, so don't say anything bad.
You hear me over the microphone? The password doesn't work for the phone. I tried it.
We hear you, David. Those 3,000 people were asked in shopping malls and other marketplaces. So they're not a random population. They were people who were more likely to be out either flaunting the rules or just doing the shopping that's within the rules, but they weren't the people who were confined at home. So there's probably an overestimate, even if you believe the test was accurate. Nevertheless, let's come back to the question of New York. Let's assume for a moment that the 20% number is correct. What we're all looking for, of course, is enough immunity in the population to believe that we've established what's called herd immunity, where so many people have effective antibodies against the virus that the virus simply can't any longer spread through the population and affect others who might be susceptible to its effects. And you generally are thought to need about 70 to 80% of the population to be infected to establish this thing we call herd immunity. And we're nowhere near that yet. And that's, of course, in the epicenter of this epidemic, and everywhere else in the country, the numbers are closer to 2 to 5%. So when Tony Fauci says, make no mistake, if you let down all of these behaviors that have been shielding us from a wider spread of infection over the next few months, there's going to be a recurrence of a recurrent increase in the number of people who are infected, the number of people who get sick.
And that's why, as you go and let down these restrictions, and as people have more contacts, testing and contact tracing become even more important, right?
Absolutely.
Because what we did was we took that R number, that reproduction number, whatever it is, by sheltering in place, we reduce that. By stopping to shelter in place, we're going to increase it. And the only way we can keep it under control by not sheltering in place is testing and contact tracing.
I think it's important for people.
Sorry.
I think it's important for people to understand that there is another step involved when you move from what we currently have, which is most of us, like me, up here in my house in the Hudson Valley, pretty much confined to one person who seems to be amiable most of the time, and that's fine. To a time when more of us are back in society and being tested regularly to see whether we've contracted the virus and should go into quarantine, because the adjacent step to identifying me and telling me to go into quarantine for 2 weeks is asking me who I've been in touch with, and not just by Zoom, but by real human contact, and may have infected. And that's going to require a whole set of caseworkers that our country doesn't really have at the moment. We're going to need people who can go and interview folks who've been identified by this mass testing, and then go to the contacts and appropriately approach them. We're going to need to harness a large cohort of people to do this kind of thing. And I think this might be a good time for us to consider some fairly radical steps that are not unlike what FDR did during the Depression. Take people who are out of work, put them to work, learn these basic social skills to do contact tracing, give them a salary, because we've got to get our money moving again. And we haven't said much about the social and economic and legal effects of all this. I know that is other nights, but it's so closely tied to the kinds of things that we as medical scientists are thinking about, that I think really has to be connected to the kinds of epidemiological protocols that we're discussing.
I don't think we fully understand the extent of the viral burden in the United States. We are 4% of the world's population, but we have some 25% of the cases. Now, we can talk about reporting, but in part because of world travel, because we got hit on both coasts, people coming from Asia, people coming from Europe. I mean, we have the highest epidemiological curves in the world.
I think it's interesting to think about that, David, because the fact that we're wealthy and move around and you're out there, we're very social, that's may make us very different than people in Africa and parts of South America and parts of Asia, who you might think traditionally would be the ones who get hardest hit. Now, they're going to be hit later on, but the virus has to come to them. They're not out getting the virus. They don't have as many visitors coming to their countries. And our affluence has probably hurt us here. You look at where is this epidemic been felt most acutely? In Europe, in wealthy parts of Asia, and in the US. And that there's a sociological aspect to that's important.
So what I am hearing, I think, from both of you is an answer potentially to probably the most common question that I'm getting on my Google Doc here, of questions that are being submitted by people who are viewing this. And that is, what is it going to take for us to be able to come out of our sheltering? And what I think I'm hearing, David, you saying very clearly that we have to have extraordinarily expanded testing capacity and contact tracing, or we are going to be back in shelter again as we are now. Is that accurate?
We lost you again, David. We've lost you. So let me just say one thing in addition to what you just said. What you said is very true, but there's one other thing we as a society are going to have a very hard time coping with, and that is it's not just having these extreme measures of either sheltering in place or having a lot of testing and contact tracing. We're going to have a change in habits. No handshaking, wearing of masks, gloves, being conscious of contacts and ways you might transmit the virus. And this is going to be in place for quite a while until we have one of roughly three things. Either we're going to have good treatment that'll mean you don't usually die if you get this infection, and people are going to be less risk averse. They're just not going to care about wearing a mask if they can be treated with an effective drug. Second, we might have herd immunity eventually. That will entail, at least under current circumstances, a lot of loss of life before we get there, in my opinion. And the third is, of course, the most obvious, and that's to have a vaccine. And I do think that there's a reasonable chance we'll have a vaccine, but everybody keeps saying it'll be in 12 to 18 months. And yet, of course, we don't know whether we actually will have one, number one, how good it is, or whether it'll be that quick. If we look at the history of production of vaccines, there aren't very many vaccines that have been brought to market into a wide population in such a brief period of time. We do have much better technologies. There are vaccine candidates that have been put into people very, very swiftly after this virus was identified. So I'm encouraged by that. But we also know that if we have to use a traditional kind of vaccine, that it could be 2 or 3 years before we have one. And that's a very long time in modern life.
Can I ask a question that one of our viewers sent in, which is, I think, an interesting one, which is there any evidence that the coronavirus is mutating in the way that influenza mutates, which would put us in a position of having to create a new vaccine every few years?
Maybe I should answer that as a virologist. These two viruses are quite different. For one thing, the coronavirus chromosome is one piece of RNA, whereas the influenza chromosome is in 8 pieces of RNA, and those pieces can get shuffled very, very easily. And furthermore, mutation rate for the two viruses seem to be quite different, with the influenza rate appreciably higher than the rate for the coronavirus. So we know that mutations occur. In fact, one of the reasons we can do such interesting studies of the evolution of this virus, how it came from SARS-CoV-1 to become SARS-CoV-2, how we can distinguish not just strains that were in Europe that came to the States or strains that were in China that came to the States, but how within a single community, based on just 1 or 2 changes in the 30,000 nucleotide genome, you can tell who got the virus from whom. That's going to be very important very shortly, because we're going to be asking, I predict within the next several months, whether someone who seems to have the appearance of a second infection with SARS-CoV-2 has had a recurrence of the original infection, or has been infected by somebody else who has a slightly different strain of virus. And we can tell from these subtle differences in the genome which is the case. And I think people have to reflect on the power of modern science to be able to distinguish between an example of the virus that has only 1 or 2 differences in the 30,000 nucleotide chain.
So Harold, a number of questions are coming in about an understandable anxiety about the reliability of tests. Not just the RT-PCR test for the presence of the virus, but the serology tests. And yes, how are we going to get to a point where we have confidence that we have a test that will give us a highly accurate reading?
Well, let me first take the 2 kinds of tests separately. Tests for the virus are most likely to fail because there simply wasn't enough virus on the swab or the sample that was used for the examination, and that's just a biological variation that we're going to have to deal with by sampling people twice or three times rather than just once, and because people will be infected and the virus won't be detected for reasons that are understandable. Those tests in general are quite reliable with a pretty high rate of a pretty low rate of false negativity and extremely low rates of false positivity. With respect to the serological tests, the issue is more difficult. And you can imagine, without being a sophisticated scientist or an immunologist, why that might be. I've already mentioned that there are lots of other coronaviruses. If you're testing for coronavirus antibody in the blood, which is what you're usually doing with a so-called serological test, if you've been infected with one of these other coronaviruses that cause the common cold, and the test that you're using is not designed to be specific for this current virus, SARS-CoV-2, it's possible to have a false positive because you're detecting the residue of an infection that occurred years ago with one of these fairly benign human coronaviruses. And for other reasons, false negatives are also possible. So it's going to be important to have careful regulatory oversight of the tests that come into common use. I understand why federal agencies might have been eager to get some tests into play and curtail some of the usual regulatory oversight, but eventually a few tests will emerge as the most effective. It's quite easy to construct samples that we know do and don't have viral antibodies to be sure the tests are being done properly and give a very low number of false negatives and false positives. But I think it's also important to stress, and I'd like to do this repeatedly, that when you're testing for antibody against the virus, you may have a very reliable test that tells you whether or not that person has been infected with SARS-CoV-2 in the past. But it doesn't tell you necessarily whether that person is truly immune to reinfection, because unless you have a way to show the antibodies are actually neutralizing the activity of the virus.
Sure. But all our experience, thank you. I mean, all our experience, and again, I agree with Harold, that we don't know the clinical question about clinical immunity. There's certainly a reason to believe, right, that neutralizing antibodies may confer. We don't know how long, but certainly our past experience.
We went over this when you were having trouble with your your microphone. Yeah.
But David, maybe you can speak to the question about the reliability of the tests. I mean, what role do the regulatory agencies have in ensuring that, you know, when an employer is testing their employees, that the test is good, reliable, and will give accurate results? Oh no.
Who is the regulator here?
I don't know, but whoever is the regulator, we're not getting it properly.
You look good. We can't hear what you're saying.
Hold that thought, David, until we figure out how to get your sound
Maybe
back.
Write it down and hold it up to the camera.
So Harold, you know, this profoundly important question about distinguishing people who have antibodies that are simply against against the virus, but not neutralizing, and those that are neutralizing. How are we going to be able to tell the difference in a population study?
Yeah, that's a really good question. I think it's going to be based on if we can figure out which proteins in the virus are targets that result in neutralization of the ability of the virus to infect, if there's a good antibody against them, then we'll have a pretty good idea. Because, well, for example, I mentioned earlier the spikes on the virus surface that mediate the interaction of the virus with the host cells. We actually know already, based on work done with the SARS virus, that exactly which part of that protein is required for the virus to enter cells by binding to a cell protein that is on the surface of the cell. And we will have, I think, very soon, tests that are based on the ability of that patient to have produced an antibody which attacks that part of the virus surface, and those will be probably authenticated as tests that are neutralizing. There may be other antibodies that are neutralizing as well. There's a lot of interest in some of the other surprisingly many proteins that are made by this virus, and some of those proteins help to make the virus structure intact. And you can imagine that an antibody that attacks those proteins would be a legitimate target for an antibody that basically blocks the capacity of the virus to enter new cells.
Yeah. So are you optimistic about a vaccine?
I think we'll have one eventually. I think it may take longer than 12 months to 15 or 20 months. But I think it's reasonable to be somewhat optimistic. The problem, of course, is that even a year and a half is a pretty long time in our society. And given the effects of this epidemic on our social lives and especially on our economy, I don't think we can't wait that much time. Even if we have a good outcome, a good vaccine, a year and a half from now, we still have a national emergency because we have to get back to doing some of the things that we do. This is the effects on mental health, the effects on people's income, their attitudes toward life. It's not my latte I'm worried about. I'm worried about collapse of society in a much more dramatic way. And we've got to figure out ways to live with the virus still circulating, many people still susceptible to virus infection, in a way that's compatible with at least a reasonably effective economy. And the country will come back eventually, but this whole episode is going to have an effect on our country that is similar to what happened during the Great Depression in the 30s.
So what do you make of the experiment that they're doing in Sweden right now, where they have made the decision not to sequester, to basically allow society to continue, but to sequester those who they identify as vulnerable, which are individuals over the age of 65 and those who have underlying health vulnerabilities? What do you make of that experiment?
I think it's something we deeply need to understand much more profoundly. That means, of course, going in there and doing the things we've been talking about, testing everybody for virus, testing them for serological responses, the evidence that infection has occurred. It is easier from a political point of view, initially at least, to say everybody should sequester inside, not knowing who's really vulnerable. I'm sure that in Sweden, I mean, we know they have cases, they have people who died, but they minimize the risks in a way that probably would be seen acceptable to most people. And I think we simply need to do some epidemiological and sociological studies to find out how people were reacting. We can say that they didn't apply all the strictures that have been applied in the US and in some European countries, but we really need to know what people did as opposed to what they were told to do, because I think we may find that you can't sit in Sweden and be oblivious to what's happening in the rest of the world. They have newspapers, they have TV shows, they go on the internet, and I suspect that a lot of people, we see movies on the night on NewsHour of folks wandering around on dockside in Stockholm, and it all looks jolly in the sunshine. But I suspect that people are being a lot more cautious than we think, and that they're obeying social restrictions that are not usual.
Yeah. So one of the statistics that I've heard is that the actual death rate among individuals who do not have some underlying health um, problem is very, very low. Is that something that is likely to hold up?
Yes,
we don't know. We don't have enough data, Shirley, to say what the death rate is. In the last day or two, there are increasing reports that the death rate is maybe 1.5 times to 2 times the number of deaths that we think. So it is just too early. The reporting has not yet been scrutinized to know what the actual death rate is.
Yeah, that is absolutely correct. But I also think that Shirley's right that the evidence is pretty strong to suggest that the kind of factors you're considering, which is that people, I have acquaintances who have underlying conditions who died fairly suddenly and would have normally been considered to have died of their underlying cancer or heart disease, who were found to be positive for the COVID virus and now are being called COVID deaths and probably were COVID deaths. And we see that by looking at the difference between expected number of deaths and the real number. And but I still think that comorbidity is a huge issue, and we don't really understand why that is. I mean, it's also true in influenza and other infections that have relatively low mortality rates.
So we're very close to the end of our time. So I want to ask a question that has come across from our viewers, which is, what do we need to learn from this one to protect ourselves more effectively from the next one? And the additional part of the question is, what are the other pathogens out there lurking that we should be paying a lot of attention to?
Okay. David, can you want to try to answer that?
Yeah. I think that one of the things we learned is that leadership is very important. And that certainly from the beginning, when there is intelligence of a significant threat out there, that you have to let the experts from the beginning take center stage and call it the way it is.
Yeah, I think obviously having a strong government response to an outbreak is critically important, and we would all endorse the idea, at least all of us on this call, I think would endorse the idea that we've had abysmal leadership from the top on this occasion. But there are some specific scientific issues that this pandemic brings up. Perhaps the most fundamental is taking more seriously the kinds of recommendations that the scientific community and the public health officials always make after a pandemic. And I think because of the magnitude of this one, as opposed to the relatively modest impact that even the influenza epidemic of 2009, and certainly outbreaks like the Ebola outbreak and SARS and MERS, which had pretty marginal effects on the US, produced. I think this one will really make us reflect on the wisdom of an investment of even hundreds of billions to prevent the kind of disaster we're seeing, which are costing us trillions. The second thing is that we are developing tests with new methods and possibly vaccines and therapeutics with new methods that should inspire us to have platforms for developing new tests that are rigorous more quickly and for developing new ways to control outbreaks, regardless of what the organisms might be. I think it's very difficult for us to say what the next pandemic is going to be. It will probably be a virus, but we're pretty well equipped for identifying viruses quickly. In general, we seem to be encountering viruses that belong at least to viral classes we know something about. That's good. It's a good argument for investing in fundamental virology, just as we were prepared for HIV because we'd invested hundreds of millions of dollars in the study of that virus class, the retroviruses. Before, here too, we had invested a fair amount of money in the study of coronaviruses, which were known to be widespread in the animal kingdom, and that did help to prepare us, even though it doesn't feel like we were prepared, but we at least knew enough to recognize this virus and many of the things it does. And we're remembering that the only drug that's shown any signs of efficacy so far, remdesivir, is a kind of an agent which was developed to interfere with the enzymes required to copy the RNA chromosomes of RNA viruses of animals. I'm going to stop there. I see time's getting late.
Right. CDC used to be the gold standard, the agency that we all look to. And what we see is that investment not only at CDC, but in state and local public health agencies have taken a hit over the last decade or so.
Yeah. So leadership, public health, basic science, all of those are the things that we're going to need to invest in going forward.
And sure, if I can just add, there's one other point, right? And that is we also need deep wells of patients right now. Right? I mean, there is no doubt that as we lift these restrictions, right? If we increase our contacts, no matter what the testing, no matter what the contact tracing is, if we go back to having the same number of contacts that we had in our cities and in our closed spaces, we're going to have a very significant another wave. We need to have deep wells of patients.
Thank you both. And David, I am so sorry that we had trouble with your audio. It was wonderful to see you and hear you when we could. And Harold, thank you so much for being a part of this. And perhaps on the note that we have been talking about for the last minute or so, perhaps it's appropriate in bringing Biddy back to conclude this session to point out that 5 of the 6 nations that have overcome this pandemic most successfully in the world are 5 of the 6 are led by women. And with that, Biddy, I'm going to turn it back to you.
Well, Shirley, that puts a lot of pressure on me, but also on you. Thank you so much. I want to apologize to you 3 and also to all of our audience for the technical difficulties. I'm sure it was terribly frustrating, and I'm really sorry. But thank you so much for being available for all that we learned tonight. And thanks to the audience for your attendance and your questions. There will be 2 more programs next week, which we'll make sure you're aware of, and all technical difficulties will have been fixed. And David Kessler will have any opportunity that he wishes to have to speak at great length on every subject that came up tonight. We will make time.
I don't think good hands with Harold and Shirley.
Thank you all so much. Good night.
Okay. Bye, Betty. Thank you.